I'd like to start by asking if any of you face challenges in your work similar to the questions that are written on this slide. For instance, during the verification of synthesis, there will be a large number of samples that may make it difficult to finish the analysis efficiently. Additionally, during the subsequent purification process, it can be challenging to explore the conditions for separation. Also, after purification, the desaltation and refining treatment can also be quite cumbersome. I believe that there are various challenges and issues depending on the specific circumstances in your field. In this seminar, we will address these challenges and concerns by presenting solutions to enhance the efficiency of purification process. Specifically, I'll be introducing two systems and a software set from our company, Shimadzu Scientific Instruments, that can help facilitate this improvement. First, I would like to discuss the LCMS purification system and OpenSolution. OpenSolution is a software designed for environments where multiple users share a single device, facilitating easy use of analytical LCMS and preparative LCMS. By following administrators to manage the equipment and methods, multiple operators can perform analyses and interpretations without worrying about the intricate details. Additionally, by placing restrictions on equipment management and method editing, we can prevent potential issues and support the efficient operation of the system. Now, let's break down the workflow of the purification processes in drug discovery into 4 steps. 1, analysis for synthesis verification and condition optimization for purification. 2, scale up from analytical to preparative. 3, execution of purification. And 4, purification processes such as desaltation and powdering of the purified substances to proceed to the next stage. OpenSolution significantly contributes to improving the efficiency of operations, particularly in the transition from analysis to purification. Specifically, the 4 challenges that OpenSolution addresses are highlighted on this slide. Now let's dive a little bit deeper into each of these challenges. We'll start with our first challenge, which is the desire to quickly finish the analysis. And this can be addressed by using OpenSolutions, enabling intuitive analysis and interpretation. With OpenSolution, it's possible to register analyses on a single screen. After logging in, operators can execute the analysis through 4 simple steps. 1, select the vials. 2, select the method. 3, input the target mass. And 4, click Start. Additionally, during the sample registration process, users can check the estimated time until their sample analysis is executed. This feature makes it easier to plan when multiple users are operating the system, reducing potential waiting times. In the analysis results screen, by clicking on the position of the vial set in the top right corner, the corresponding analysis data will be displayed. Users can click on the displayed peaks to view the corresponding mass spec and UV spectra, allowing for intuitive analysis of the desired peaks. Furthermore, the system supports automatic generation of analysis reports, enabling efficient verification of the presence of target components within the analyzed samples. Now let's go over our second challenge, which pertains to equipment management. This is also supported by OpenSolutions through its feature of automatic conditioning. OpenSolution enables automatic startup, operation, and shutdown through its reservation event feature. The main scenarios for utilization include the 3 types, also known as startup, conditioning, and shutdown. With startup, users can designate specific days and times to automatically execute batch analyses. By scheduling the startup for early mornings on weekdays, the system can complete conditioning of the equipment by the time operators arrive for work, allowing them to begin analysis immediately. With the conditioning, this function automatically executes solvent switching and methods for equilibration based on the registered analysis methods. As a result, operators can obtain stable data without having to worry about the conditions of the previous analysis or the state of the equipment. And in shutdown, If the equipment has not been used for a certain period of time, this feature automatically performs flow adjustments and shuts down the equipment. This prevents mobile phases from continuously flowing and from the equipment from maintaining operational unnecessarily. When multiple users share a single device, determining the timing for shutdown can sometimes be challenging. However, with this functionality, the equipment can automatically stop if analyses are not conducted for a certain period of time, ensuring efficient equipment management without requiring deliberate shutdown actions. Additionally, the reservation event feature allows for the execution of system suitability testing, or SST. With Open Solution, users can define the retention times, peak areas, and heights based on specified analyses, enabling assessment of whether the equipment is in a usable state. By scheduling the SST to run at specified times each day, the results can be automatically organized into lists and graphs, allowing for easy monitoring of the system's condition over time. For instance, as demonstrated in the graph on the bottom right, it becomes easy to observe time-based changes and any anomalies at a glance. Moreover, users can simultaneously review multiple systems, significantly alleviating the burden on equipment managers. All right, we're halfway through, and now we're going to tackle the third challenge, which pertains to concerns about the status of analyses, such as wondering when the analysis will be finished or if it has been completed already. By utilizing Open Solutions, users can perform analysis from their office, even when they're not physically present in front of the analytical equipment or control PC. This capability allows operators to make effective use of their waiting time, contributing to enhanced productivity. By establishing a network as illustrated on this slide, Open Solution contributes to improved productivity. Data acquired from the equipment can be centrally managed on a server, allowing data analysis to be conducted from separate office PCs or individual operator computers. Not only can users check the status of analyses during registration, but automatic email notifications are also sent to the responsible personnel upon completion of analyses. Through the link in the email, users can easily check the data and access analysis features from their offices. This functionality eliminates the need to wait for a computer to be able for analysis when multiple users are involved, thereby enhancing operational efficiency. And lastly, the fourth challenge relates to the difficulties in exploring separation conditions for purification. By utilizing the automated scale-up for analysis and purification feature in Open Solution, significant improvements in operational efficiency can be achieved. In the field of drug discovery, it's common to encounter cycles similar to those shown on this slide. However, in environments where small quantities of a wide variety of compounds are synthesized, the purification step can become a bottleneck. For instance, while parallel automated synthesis devices can allow for the simultaneous synthesis of multiple samples, the purification process often has to be carried out one sample at a time in sequence. In such cases, there's a concept of applying focus conditions, calculated from the retention times of standard analytical conditions, directly to purification without actual measurement, and doing so by loading multiple samples at once to enhance efficiency. The feature that enables this shortcut is Open Solutions' automated scale-up for analysis and purification. The automated scale-up feature for analysis and purification, which we call ASAPrep, assists in the optimization and automation of scale-up methods from analytical conditions to purification conditions. During screening analysis, multiple analytical conditions are tested simultaneously to explore optimal separation conditions. For example, samples can be analyzed under acidic and basic mobile phase conditions to determine which setting is more suitable for purification. Against the screening results, the software employs predefined criteria to assess the analytical LC-MS data for potential coelution. It automatically determines the mass spectral purity of the target compound and checks for any impurities eluting near the target compound. If the peak of the target compound is sufficiently separated from other impurity peaks, there's a high likelihood of good separation, even when scaled up to purification conditions, resulting in a green Go designation. In this case, optimized purification LC gradient conditions are automatically generated. Conversely, if impurities are present near the retention time of the target compound, or if the target compound shows incomplete separation, it's likely that separation at the purification scale will also be inadequate, resulting in a yellow designation. Should the target compound not be detected, a red designation is given. For samples with a yellow designation, improvement in separation must be achieved by modifying conditions such as columns, mobile phases, or gradients to recover the target component in a pure form. In environments where the number of chromatography experts is limited, the automation of green designations allows for efficient task delegation, with only yellow or red designations requiring the expertise of chromatography specialists. This approach facilitates effective workflow optimization and contributes to enhanced overall efficiency for the entire team. Here, we have the optimized purification LC gradient conditions, specifically the initial gradient concentration and slope, that are automatically determined based on the retention times of the target compounds obtained from the screening results. These purification gradient conditions are established using pre-registered formulas within the software to ensure that the target components elute at the specified time frames. This slide illustrates an example where the target components from all samples are designed to elute around 4.3 minutes at the purification scale, correlating the retention times from screening analysis with the initial gradient concentrations for purification. It's easy to envision how successfully separating the target components and ensuring their elution at designated times can significantly alleviate the burden placed on operators. Now, allow me to explain how this workflow is executed in OpenSolution from the operator's perspective, outlining the sequence of operations. First, we begin with the screening analysis conditions. As mentioned earlier, operators can execute this process simply by performing the following operations on a single screen. 1. Specify the vial. 2. Select the method for screening analysis. And then 3. Register the target mass. It's common to set up two conditions for the screening analysis method, one for acidic and one for basic conditions. The results from the screening analysis can be compared side by side on a single screen for each sample when conducted under the two conditions of acidic and basic. Additionally, the color-coded assessment for scale-up can be easily reviewed for multiple samples and multiple conditions all at once. If a yellow or red designation is given, the reasons for the assessment will be displayed, which makes it easier to formulate the next steps even if the outcome isn't green. Next, by loading the scale-up assessment results, the optimal gradient conditions are automatically set. The display is organized based on the color-coded assessment results, allowing the operator simply check off whether or not to proceed with the purification and register their choice to execute the purification process. And finally, regarding the confirmation of purification results, operators can easily analyze the outcomes with simple operations. By selecting the sample vial set in the top right corner of the screen, the test tube containing the purified fraction is automatically highlighted. Furthermore, when the operator selects the highlighted test tube, the corresponding range of the collected fraction is indicated on the chromatogram, accompanied by the mass spectrum of the fraction. This allows the operator to retrieve only the test tubes that contain the target components at high purity, progressing to the next steps of purification and powdering. Such straightforward operations enable even those who are not chromatography specialists to successfully carry out purification tasks. Additionally, this standardization of workflows makes it easier for multiple users to collaborate effectively. Now, let me present some practical examples of application. First, we have a case demonstrating the application of the ASA prep feature to low molecular weight pharmaceuticals. The screening analysis results for all samples received a green designation, indicating that high purity purification was successfully achieved using the automatically generated methods. By utilizing the ASA prep feature integrated into OpenSolution, anyone can carry out high purity purification with simple operations, leading to significant improvements in operational efficiency. We do have solutions for such cases as well. By utilizing the software Lab Solutions MD, which supports efficient method development for analysis, you can streamline the development of methods aimed at separation improvement. Lab Solutions MD works in conjunction with a method scouting system that allows for solvent switching and column switching, facilitating initial screening to explore suitable combinations of solvents and columns for separation. Once the solvent and column have been determined, gradient optimization can be performed with simple operations. During gradient optimization, the application of AI algorithms for automatic optimization under reverse phase LC conditions can allow for significant reduction in the time and effort required for method development, independent of the operator's experience. Now, let's take a look at the workflow when the screening analysis results are designated yellow. Here, we have an application example involving synthetic peptides, where the sample mixture contains the target component, paratoluene. The screening analysis results indicated that the target component co-eluted with impurities, resulting in a yellow designation. In response, by utilizing Lab Solutions MD to perform solvent screening and gradient optimization, the separation between the target component and the impurities was successfully improved. After conducting a load evaluation at the analysis scale, purification and scale-up were executed. Furthermore, from the reanalysis of the fractions post-purification, it was confirmed that high purity purification of the target component was successfully achieved. As illustrated in the workflow on this slide, even when the screening analysis results indicate a yellow designation, meaning insufficient separation, you can utilize Lab Solutions MD to improve your separation. By executing purification based on the resulting enhancements, high purity products can be attained. I'd also like to present two additional examples involving cyclic peptides. The first example is similar to the previous slide, where the screening analysis indicated that the target component co-eluted with impurities, resulting in a yellow designation. Again, by utilizing Lab Solutions MD, separation improvement was achieved, allowing for the recovery of a high purity target component. The second example involves a case where the UV chromatogram from the screening analysis displayed a single peak, appearing to indicate a green designation. However, by examining the separation conditions using Lab Solutions MD, we were able to separate minor impurities that were hidden beneath the main peak, ultimately recovering an extremely high purity target component. This demonstrates how Lab Solutions MD is a valuable tool, depending on the required purity. However, the choice between applying OpenSolution ASA Prep for purification or utilizing Lab Solutions MD for optimizing separation conditions needs to be made according to the throughput and purity demands specific to the drug discovery purification field. Now, let's shift gears for a moment because I'd like to introduce the Online 2D Purification System UFPLC and its dedicated software, Purification Solution, which can significantly streamline the post-purification processing tasks. By using this system and software, we can expect improvements in operational efficiency, particularly in the workflow from purification to powdering in the purification processes. As shown on this slide, do any of you encounter challenges or concerns related to the purification and powdering stages? Let's begin with the purification process. Depending on the intended use for the next steps, it's often necessary to remove salts that are contained in the mobile phase used during purification. Additionally, in cases where acidic or basic compounds need to be recovered in their free forms, Many people resort to solid phase extraction columns or liquid-liquid extraction using separatory funnels. However, performing purification tasks manually can be cumbersome, and concerns about sample loss during processing may arise. Now, regarding the post-purification processes, it's often necessary to concentrate or powder the samples for subsequent steps. Depending on the conditions during purification and how the samples are processed, there may be significant amounts of water in the sample solvent, leading to extended drying times. In such cases, you might be looking for ways to reduce drying time or to recover high concentration samples from small volumes of solution. The online 2D fractionation purification system, UFPLC, can automate the entire process from fractionation, concentration, purification, elution, to recovery, all in one system, as shown in this slide. First, let's take a closer look at the differences between conventional purification LC and the online 2D purification system, UFPLC. In conventional purification LC, samples that have been separated in the column are collected directly using a fraction collector. In contrast with UFPLC, as demonstrated on the previous slide, after being separated in the column, the separated components are directed into a secondary column called a trap column, where they're retained. Following this, desalting with ultra-pure water and free base treatment with ammonia water are carried out. And finally, the components are recovered using an organic solvent solution, such as acetonitrile. The trap column can accommodate up to 5 units, allowing separate retention of 5 different components in different trap columns. Moreover, the process of separation, followed by concentration, can be repeated multiple times, enabling the retention of the separated components in the trap columns. This allows for the concentration of target components on the trap columns, which can ultimately be purified and collected all at once. Now, let's delve into 3 examples demonstrating the efficiencies gained by utilizing UFPLC. The first example involves the recovery of components after removing non-volatile salts from the mobile phase. After the fractions containing the target components are introduced into the trap column and collected, they're washed with a solvent that does not contain salts, allowing for the elution from trap column while effectively removing the salts that were necessary during separation. In the case of purifying ibuprofen using a mobile phase that contains non-volatile salts, such as ammonium dichloroacetate, conventional LC methods would collect ibuprofen along with the salts derived from the mobile phase. However, with UFPLC, ibuprofen can be collected in a state free from the salts. In HPLC analysis, it's common to construct an analytical system using phosphate buffer solutions. However, during purification, non-volatile phosphates can become part of the recovery, complicating the post-processing steps. In such situations, it would typically be necessary to substitute with conditions using volatile salts, such as formic acid. But by utilizing UFPLC, that allows for this process without such complications, making it adaptable for purification. The second example involves the removal of TFA derived from the mobile phase, allowing for the recovery of basic compounds in their free base forms. Basic compounds, when collected along with counter ions or ion pair reagents such as trifluoroacetic acid, or TFA, that are present in the mobile phase, that can potentially affect the results of subsequent assays. With UFPLC, after capturing the target components in the trap column, these salts can be washed away, enabling the recovery of the target components as high-purity, free base forms. This slide presents the analytical results obtained through ion chromatography for a TFA solution containing the basic compound propranolol after undergoing free base treatment. Compared to untreated samples, the recovery conducted using UFPLC shows a significant reduction in peaks corresponding to chloride ions, NTFA-derived peaks. This confirms that effective removal of counter-ions has been achieved during the desalting and free base treatment processes. The final example involves shortening the recovery and powdering time for concentrated samples. As previously mentioned, UFPLC allows for the repeated injection of samples, whereby fractions containing the target components can be introduced into the same trap column. This enables the concentration of the target components within the trap column. After concentration, eluting the target components from the trap column with an organic solvent can significantly reduce the time required for powdering. In a comparison of powdering times for a fraction containing 100 milligrams of ibuprofen using a centrifuge for concentration, it was found that the time taken was reduced to about half compared to using a conventional purification LC system. By utilizing this repeated injection feature, not only can relatively large quantities of samples, up to approximately 100 milligrams, be purified more efficiently, but it is also adaptable for concentrating and recovering trace components. Next, I'd like to introduce the dedicated software for controlling the UFPLC, called Purification Solution. By using Purification Solution, you can easily execute the flow of processes from separation to concentration to purification to elution to recovery, as outlined in the previous slides. During execution, you can monitor the status of the equipment from a single screen. The peaks in the purification chromatogram, the status of component introduction into the trap columns, and the colors of the collection containers are linked, allowing for an instant understanding of which components have been introduced into which trap columns and subsequently recovered into which test tubes. Furthermore, even for multi-step processes, pre-prepared template method files eliminate the need for complex settings, enabling anyone to easily execute purification and separation tasks. Moreover, this system and software can be used not only as UFPLC, but also as a conventional purification LC. Switching between the two modes can be easily done through the software, allowing users to flexibly operate either the conventional purification LC or the online 2D purification system, UFPLC, according to their specific needs. This adaptability enhances the system's versatility and usability in various purification scenarios. Now, we've covered both Open Solution and ASA Prep, along with UFPLC and Purification Solution. This is going to be the final slide of our webinar. We've conveyed that Open Solution and ASA Prep can streamline the workflow from analysis to scale up to purification, while UFPLC and Purification Solution enhances the efficiency of the workflow from purification to recovery. Additionally, the transition of methods from Open Solution to Purification Solution can be easily accomplished, enabling a seamless connection between these software platforms and covering the entire workflow from analysis to purification. Furthermore, during the stage of analyzing method conditions, efficiency can be enhanced by utilizing the analytical method development support software, Lab Solutions MD. In this way, Shimadzu Scientific Instruments supports the efficiency of your purification processes with a broad range of systems and software. We hope that the solutions introduced today will be of help in improving the efficiency of your operations. I'd like to thank all of you for attending this presentation today. Again, under the resource list, you can find more information about our analytical solutions. If you haven't had a chance, please fill out the survey questions on the left-hand side of the screen to provide feedback and request additional information. I also encourage you and your colleagues to attend our upcoming webinars, which can encompass a wide variety of application areas. With that being said, have a great rest of your day.